Hello, everyone, and welcome to today's live broadcast. The Genes and the Beans, high-quality reference genomes provide tools for coffee breeding improvement, presented by Susan Strickler, PhD. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Dovetail Genomics. Dovetail Genomics is transforming genomics by making long-range genomic information readily accessible to all. The company enables researchers and clinicians to solve complex problems involving de novo assembly, structural variation, cancer research, and more by providing a comprehensive view of the genome. Their proprietary proximity ligation and informatics approaches simplify genomic discovery by integrating the highest quality long-range genomic information with next-gen sequencing output. For more information, please visit dovetailgenomics.com. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce today's speakers. Dr. Xuan Ho, who will be providing an introduction to Dovetail technology. Xuan, a former Dovetail Genomics customer who led the quinoa genome informatics, recently joined the team as bioinformatics production manager. Xuan received his PhD in computational biology from the University of Sydney. Prior to Dovetail, Xuan served as the NGS bioinformatics officer at the Australian Genome Research Facility, did his postdoctoral fellowship, and was lead genomics and bioinformatics research scientist at KAUST in Saudi Arabia, and most recently, the senior manager at A*STAR in Singapore. Dr. Strickler is the director of the BTI Computational Biology Center at the Boyce Thompson Institute. Dr. Strickler is the lead bioinformatician of the Arabica Coffee Genome Consortium, and her research aims to produce genomic resources to be used in comparative genomics, gene family analysis, and other studies to better understand the evolutionary history of Coffea arabica and to develop tools for the improvement of coffee through breeding. Please join me in welcoming Dr. Ho and Dr. Strickler. I will now turn the presentation over to them.
Thank you, Judy. Well, welcome to this webinar, and we're very happy for you to be able to join us over the internet. So, just a very, very brief introduction to myself. I was a previous customer of Dovetail when I was creating the genome for quinoa. At that point of time, we were looking for an effective solution to build a genome up to a chromosome scale. And Dovetail had the technology that kind of fits what we needed. So in this slide in the middle, I'll just show some stats that we actually presented at PAC 2016 a little while ago, which is also found in a Nature paper, if you're interested. As you can see, there's a improvement of N50 all the way from 2.4 megabase all the way up to 3.84 megabase. So we were very happy about this, and which is why I'm continuing my work at Dovetail to work more on genome assemblies. But this whole talk is not about quinoa, and I'm interested to tell you about the underlying science that make this all possible. So just to go forward, this is who we are, this bright-looking bunch of people. So this is what we do. So in a nutshell, we do genome assemblies all the way from library preparation. We can do sequencing of it for you. And once we have the data, we can do a full de novo assembly all the way to get an assembly. And with the data that we get from Chicago and Hi-C, we can do a scaffolding for you all the way up to chromosomal level assemblies. We understand that no two genomes are the same, and they all come with their own challenges. Therefore, it's our goal that we want to make it a more fully integrated from sample to chromosome. A very bespoke service so that you guys get what you want out of this project. As for example, when you go into a restaurant, you probably want to look at the menu. So this is just a very simple list of things that we offer. See, we actually do high molecular weight DNA extraction, and this is actually very important because it generates intermediate scale information of about 100 kb. So we'll talk a little bit more about this later when we discuss the different data set. But just bear in mind that having a good input is very important for the success of your assembly. The next thing we do is that we do library prep. We do normal library preps like Illumina, PacBio, and we also do our proprietary library preparation, in this case Chicago and the Hi-C. Later in the talk, I will discuss about the trade-off between these two different library preps and also the complementary aspects of the data characteristics. When you actually done the library prep, we can sequence it for you. We can sequence shotgun sequencing. We can do PacBio sequencing, or if you prefer to do it yourself, we're more than happy for you to do the sequencing. Once you have the data come back, and once we have it, if you have a shotgun data, we'll run it through our Illumina or shotgun assembler called Miraculous, or if you have long read PacBio, we'll put it through Falcon to get a starting point for our scaffolding. And the next step is the scaffolding, where we have the Hi-Rise or Chicago data set. This is our proprietary proximity ligation scaffolder, which we call Hi-Rise, which we use to stitch many pieces of small scaffolds into all the way up to chromosomal level assembly. And finally, before we give it to you, if you have any shotgun data that you have generated long before that you want to use and you want to waste, or you have PacBio or low coverage PacBio data, we can actually use it to gap fill the assembly, and just to the aim to increase our contiguity. Okay. With all these things, a lot of handful. So let's go into the science or the molecular part of this proximity ligation approach. Why is it important? Because we want to share with you how this can help with the genome scaffolding. In this slide, it's a little bit busy. I have a cartoon illustrating the different steps in this approach, all the way from left to the right. So from the left, you have DNA, and all the way to the right, you have data coming out from sequencing. I just want to point out right now, this applies to Chicago and Hi-C, and the Chicago and Hi-C applies to the middle part. So the middle part of this cartoon is pretty much the same for both kind of libraries. And what's different between Chicago and Hi-C is the starting point all the way to the left. For Chicago, you start from pure DNA, and that's how you start. Whereas for Hi-C, you actually start from host tissue or cell. And the difference is in the front and also at the back, which is the different aspects of data you get out. Okay, so without further ado, let's go into the science part of it. So, and let's use Chicago as a starting point. So if you follow along with me, so on the left-hand side, you see the little black, black line, and therefore you can see that there is a little 1 and 2. Okay, just gonna draw it out here for you. Can you see this? 1 and 2. Okay. All right. So imagine that this is DNA, a single strand of DNA that's about 100 kb, and points of point number 1 and 2 on this DNA is about 100 kb apart, and it represents one long strand. So what we do is that first we do for Chicago, we actually reconstitute the chromatin. We add enzymes, histones, and DNA, and the enzymes actually reassociate the histones to DNA, and DNA wraps around the histone. One good thing about this is that it happens in a very uniform fashion and rather evenly across the whole fragment, as you can see in the second part here. Okay. Once this happens, it actually allows us to go to the next step. So the histones is very important because they act as handles for the next step, which you'll understand very shortly. we actually cross-link the solution next, as you can see in this red bars between the little blue circle. So this cross-linking causes form covalent links between the histones. And as a result of this, a previously very long DNA from the first step is bounded together somehow now in a bit more of a globular form. What this means, essentially, if you can see from this illustration here, okay, is that point 1 and 2, which is previously very distant away in the DNA, is now brought together into close proximity. So this is the key part of this proximity ligation approach. Next, we actually use a restriction enzyme to cut out this DNA aggregate, and as a result of that, it causes sticky overhangs, which is very normal. And what you get out of this is that from a single long DNA fragment, now you have numerous fragments of DNAs. And the good thing of the crossing is that because the crossing was done before, it actually retains the proximity ligation information there before we do the next step. Once we have this, we actually fill the overhang at the same time. We want to be able to capture this kind of information, so we add a marker. So in this case, it's a modified nucleotide with a biotin attached. As you can see, this is this part is a little green area, okay? And this is important because it allows us to enrich for those very important fragments that you see next. Next, we actually perform ligation. Two fragments with the biotin are actually fused together. And what you might imagine is that because the fragments are held in close proximity throughout the cross-link, it's more likely to ligate fragments that are close together than some random fragments that's in solution. What that also means is that the chimeric molecule that is formed follows a very predictable statistical distribution that we will be able to use later for scaffolding. As you can see from this step here, 1 and 2 is actually fused together. So it's perfect for us to retain that. Finally, we actually fragment the DNA further, remove the cross-links, and remove the proteins. And what you get in this case over here, remove the biotins at the end, remove the proteins, and what you get at the end are fragments with biotin in the middle. This is very important because now we can enrich for those fragments with those biotins, which represents the important ligation that took place because of proximity ligations. With this, we can put it into the sequencer, and with that, we actually sequence both sides, and you can see that we actually sequence very close to location 1 and 2 in your whole genome. So, and then we put it through the HiRISE, and HiRISE uses to scaffold your assembly. And with that, with the difference between Chicago and Hi-C, so I've mentioned this whole process, I've just talked about Chicago. So where does Hi-C come in? For those of you who like to bring your genome to the next level, where you want chromosomal level assemblies, with the Hi-C, we do not reconstitute the chromatin. So actually, we fix the chromatin in vitro within the nuclei of the cell. And what this does is it retains very long-range information, much, much longer than extracting DNA. And we have seen it in cases where you actually can get full chromosome information from this, which I'll explain a little bit later in the next slide, where we look at the data characteristics. So before we move forward, I just want to kind of discuss a little bit about what you might want to consider as a data recipe, right? When you do a genome assembly, normally it's good that, or it's ideal that you have data representing the genome from different scales, all the way from short, intermediate, all the way down to long range. And just to make it clear what we mean by short, intermediate, and long, a short scale in our perspective will be something less than 100 kb. So those will cover libraries with your shotgun data, your mate pair libraries, even long read PEG bio, which is already very long, but in this case, in the whole perspective, the whole genome is still relatively short because it's less than 100 kb. Then you want to have a proportion of your data coming from intermediate scales. So this will be in the order of 100 kbs. And technologies out there you have is that's available to you in your arsenal of scientific tools, optical maps, which we also previously did for our project in quinoa, and 10x technology, and also Chicago. It's so easy now to do that. And finally, with long range, massive long range full chromosome information, right now there's only the Hi-C methodology, including our own dovetail proximity ligation approach. So you can see that with all this recipe you put together, it kind of helps you build a better genome at the end result. Okay, next step, we just want to look at the information, what kind of data you get. So right here I have two plots. On the left we have, they're the same data set, just plotted on two different x-axes. And the reason why we're looking at this is that I want to explain to you the characteristics of the different kind of data. On the left plot, you have the x-axis all the way up to 500 kilobases, and on the right you have all the way to 100 megabases. So every single dot shows the separation, and the y-axis shows the proportion of read pairs within the library that falls at any given separation. And what you can see is in Dovetail Chicago in green, you can see that it's a decay as it goes, getting bigger and bigger, and maybe all the way up to 100 kb, and you can still see there, you can still see some data about 250 kb, but majority of it is within the hundreds of kbs. Okay, and if you put that in the perspective of shotgun sequencing, mate pair, and even long reads, packed by long reads, you can see that where this intermediate scale be able to complement all this short range information. One thing to take out from this picture on the left, you can see that the decay in green for Chicago actually kind of plateaus or even kind of like decays all the way after 250 kb. You know, I'll say about 100 kb. But on the Dovetail Hi-C, we continue to see a kind of a flat all the way to the right, that we still can get information beyond 500 kb, which is why we're actually showing you the plot on the right. So in this case, the scale is much bigger, up to 100 megabases. So you can see that the characteristics of Hi-C actually goes all the way, even up to 100 megabases. So this is really how to say, encouraging, because it gives you a different mixture, instead of a recipe of different data sets, so that we can help you build the data. Just to show you just some results of what we did. So this is a project that we did with LSU, Brent Furclough. It's a bob white bird. So what we did for this project is we did a shotgun sequencing, we did a de novo assembly of the shotgun, we did some Chicago scaffolding, and all the way up to Hi-C scaffolding. And we have three different plots here. Just to explain what these plots mean. So every single, on the x-axis are the individual scaffolds, sorted from the biggest to the smallest, and then on the y-axis is the reverse of that. So you can, and the density of that, the color density within each boxes represents the information of how things are coming together, right? So you can see that the first thing that you see in mind from de novo, Chicago, all the way to the right, Hi-C, the boxes get bigger. This is very expected, because when you do a de novo assembly, the scaffolds are very small, which is why it doesn't cover a lot of the x-axis. And when we move on to Chicago, we can actually, the x-axis boxes actually get bigger. So in this case, a lot bigger. And in this case, when we actually move all the way to Hi-C, it's even much bigger. In this case, we can see here that, let me just use my pointer. Okay, this is actually going up to full chromosome scaffoldings. And one thing that I would like to point out is that if you can see these little lines here, these little lines here, these are actually what we see as characteristics of the Chicago data, that there are some scaffolds that seems to have some information of linking between another scaffolds. And what we could not resolve at the Chicago level, we will be able to resolve that at a Hi-C level, because you need even longer range information to solve that. So it's a very kind of neat project, because we, from the left all the way to the right, we went from a very tiny N50 assembly all the way to a full chromosome assembly. And what is nice is that it's also created within just 8 weeks from sample received to data. What this means in terms of genome utility is that you get your data faster, you can start working on it to solve whatever problems that you guys wanted to in the first place. This is not just birds. We were actually very blessed that we were able to also work on different genome assemblies. In this case, a reptile, it is a lizard, and the fish in the middle, and also the plant, which is a lettuce. So similarly, like what you have seen for the bobwhite bird, for reptile, we started off with a 500 kb N50. After Chicago, it went up to 50 megabases, and after Hi-C, it went up to 90 megabases. And for fish, it was 24 kilobases for a starting assembly. It's a little more fragmented in this case. The data wasn't as easy, but the Chicago actually saved that, because we could bridge this 24 kb for fish. After Chicago, we got 12 MB megabase, and when we add Hi-C, it was able to rescue that and 41 megabases. Same for plants, from 500 kilobases up to 6.5 after we add megabases, after we add Chicago, and all the way up to close to 300 megabases. This is at some point, this is statistically not even exciting, because we are reaching a full chromosomal level. So this is just to illustrate what is the utility of this technology that we have here. As far as polyploids, we were able to work on this project where we did a de novo sequencing assembly. We did Chicago and Hi-C for this tetraploid plant, the scantus. As you can see on the left-hand plot, it's beautiful. We could achieve a chromosomal level assembly. But more importantly, for the science part of it, from what we learned from the PI, Macanthes, it's separated from sorghum about 6 million years ago, and there seems to be a duplication event, or we believe there was a duplication event about 4.5 million years ago. So when we do a comparison, we have a dot plot on the right between Macanthes on the y-axis and sorghum on the x-axis. You can see that, wow, this could be really seen, because every pair of Macanthes genome, you could see a corresponding mapping to a sorghum. So this is very, very throughout the whole genome. So this is a great validation of their science. And just to make sure that this is a polyploid, and just to make sure that we actually got it right, they look at looking at scaffolds, actually mapping back to their linkage map, which we have created from a genetic map. You can see that there's a very nice separations of homologous scaffolds. And there's this little dot here, over here. Let me see, you can put it here. So the little dot here. You can see that these two scaffolds is linked to linkage group 1, which shows the utility of having a good starting point genome. With that, I'd like to close off this last 2 slides just to share with you guys the complementary characteristics of having Chicago versus Hi-C. What I have here in these 4 boxes, they are the same data set. Sorry, this is Chicago data and Hi-C data mapping to the same scaffold, just to illustrate our point. And on the left column, we have the Chicago data. On the right column, we've got Hi-C data. And on the x-axis is the midpoint where the pair maps onto the scaffold that we have. And on the y-axis is how far apart the reads are mapping to the scaffold. Okay. So if a pair of reads map very close together, then it will appear a little bit more on the bottom of the y-axis. If it's mapping further apart, you should see the dot a little bit more on the top part of the y-axis. And the first thing that you see that you can see from this pattern is that you got a higher density of purple dots at the bottom of the y-axis. That means that yes, as what we see from decay, a lot of the data are mapping closer together, and as you increase the distance, you get fewer and fewer dots going up on the y-axis. So this is very expected. But what we did was that we wanted to know what happened if there's actually inversion. So we artificially went into this scaffold and inverted a 50 kilobase contig within this scaffold. And now what you can see is that, I'm not sure whether you can see it with your eye, but I'll just help you there. So can you see this pattern? We can see this displacements of read pair is actually raised. So it means that they are mapping slightly higher than it's normally seen. The human eye is normally quite good at picking out this, but imagine looking at it throughout the whole genome. I wouldn't want to be doing that. I'm sure there's a better way to do that, and that's where the algorithm comes in. And what you can see in Chicago is that it can identify this kind of misplacement. And because this is a 50 kilobase pair inversion, you don't see that at a Hi-C level. And I'll just advance to the next slide so you can kind of see where the breakpoint is. You don't see the same kind of information at a Hi-C level, which means when you go into Hi-C, you have to assume that your data is actually, your input sampling is actually very good. Which kind of like to close my talk in the sense that we want to, the intermediate scale information that you get from Chicago data is especially useful if you have a very fragmented starting assembly. Because you will be able to go in and find out regions like this and try to correct them. Unfortunately, we won't be able to correct this information if you jump straight into the Hi-C with a very fragmented assembly. This is just my last thing that I'd like to highlight. And thank you everyone for listening to us. And I'd just like to let Susie talk about her project. Thank you.
Okay, thanks a lot for that. So I'll talk a bit today about some work that we've been doing with our coffee genomes, and I'm speaking on behalf of a large consortium of people, which I'll talk more about at the end. So I'm sure I don't need to make the case that coffee is important, and so many people can't get through their day without a jolt of caffeine. But there are some other reasons coffee is important as well. So it's one of the world's most valuable agricultural commodities. This is especially true in developing countries where coffee is a top exported good. And many commodities are traded at a higher level, but they aren't as valuable. It's also thought to be the largest source of antioxidants in the Western diet with many possible health benefits. So it can reduce the risk of things such as some types of cancer, type 2 diabetes, and dementia. There are many economies that are driven by the production of coffee. And as you're probably aware, it's also threatened by climate change. So you can see in the map here some of the regions that are the top producers of coffee, two different types of coffee. There's Robusta and Arabica, which I'll talk more about. And they're all more in the tropical regions, which are known to be will be affected by climate change. So for example, a study finds that Ethiopia, which is the world's 5th largest coffee producer, could lose up to 60% of its suitable farming land by the end of the century. There are 3 main species that are used for drinking coffee, but the actual coffee genus has over 120 species. So they're native to tropical Africa and Asia, and they've all evolved rather recently on the evolutionary timescale. So you can see here the 3 different species that are used mainly for drinking coffee. So coffee arabica would be the main one. So this would be your drip coffee, your espresso, your lattes. This accounts for about 75% of coffee production. Then the second highest production would be from coffee canephora, which you can see here in the middle. And it accounts for about 20% of production. And it's thought to not taste quite as nice, and it's more frequently used for things like instant coffee. And there's also a very small amount of coffee liberica that is used for coffee production to drink. So that accounts for less than 2% of production. The coffee bean, which you're probably well familiar with, is actually the seed of coffee. So you can see here in the top left of the slide the coffee cherry. So these are the coffee fruits. So these start green and they turn red when they're ripe. And then they're harvested and they're dried. And the outer husks are removed to result in the coffee beans. And then these beans are roasted to get the coffee that you would drink with your morning breakfast. There are a number of things that can go wrong here. So you can see in this poster that they can be affected by wetness whenever they're drying. They can be over-dried. They can be crushed. So whenever you're thinking about coffee production, you don't only think about the actual genetics of the coffee and growing the coffee, but then there's this whole post-harvest step that comes in that can affect the coffee quality. So I'll be focusing on 3 main species today that we've done a number of genomic studies on. So these are coffee genoides, which you can see on the top left, coffee canephora, which is the top right, and coffee arabica. So arabica is derived from a cross that happened millions of years ago, so relatively recently on the evolutionary time scale, between 2 diploid ancestors. So Eugenoides would be the female parent and canephora is the male parent. So arabica is actually something that's known as a tetraploid. So it has both of the parental genomes. And I should say that canephora was actually previously sequenced in another study. So we've added a bit of data to that one as well, which I'll tell you more about in upcoming slides. You can see here the distribution of canephora and Eugenoides. So there's a slight overlap in northeastern Africa. And arabica is found in Ethiopia at slightly higher elevations. So for people that don't work on plants, polyploidy is kind of an interesting, maybe unexpected thing to come across. There are a lot of plants that are actually polyploid. So you can see here some of the ones you may be familiar with. So potatoes are what's known as an autopolyploid. So this means its genome is derived from a duplication of a single taxon. And then the other species that you see here are known as allopolyploids. So their genomes have derived from 2 or more diverged taxa. So for example, strawberry is actually a octoploid, so it has 8 copies of its chromosomes. Tobacco is a tetraploid, cotton is a tetraploid, and wheat is extra complicated, and it's actually a hexaploid. And most plants are also ancient polyploids, so we call these paleopolyploids, which had a duplication event a very, very long time ago, and they have remnants of these throughout their genome, which can add complication to assembly and analysis, since a lot of these regions are there in more than one copy. So the coffee genome is not super big. Here we're looking at arabica, so it has the biggest genome out of the 3 that I've mentioned, since it's the tetraploid. So it's a bit bigger than Arabidopsis, which is the model plant. This is the first plant that was sequenced. But it's smaller than the human genome and much smaller than the wheat genome, which, as I mentioned, is quite large. So why should we sequence the coffee genome? So our goals with this project are to make a template, basically, that can be used for studying genetic variation, so diversity within coffee, different coffee accessions. Diversity is really important in breeding because by looking at diversity, we can find accessions that may have resistance genes, coffee has a number of pests and disease problems that need to be dealt with through breeding. There are also quality issues, so if we can get to the genetic basis of quality, then this allows us to do more efficient breeding. So those are some of the more applied reasons for a coffee genome. And also we're interested in the evolution of coffee. So as I said, it's derived from these two progenitor species, and there are a lot of really interesting things that happen whenever there's a polyploidization event. So by having the two progenitors as well as arabica, this allows us to do some really cool stuff to look at gene loss and rearrangement and other things of interest. So with any sequencing project, there's a number of things to consider and a number of different data types that need to be incorporated to get a high-quality genome. So these are some of the different things that we considered for our project, and starting with plant selection. So that's the first thing that needed to be considered, because plant genomes can be quite complicated. So we needed to decide which plant should we sequence. So as I said, canephora had been previously sequenced. So the individual that we sequenced was actually the same one that was published previously, and we've added more data to that to try to improve the assembly a bit. So canephora is a diploid, as I said previously, but the one that we sequence is a monohaploid. So this means that there should be no heterozygosity whenever we assemble the genome and do downstream analyses in this particular individual. Eugenoides is a diploid as well, but the one that we used is heterozygous. So this one is not a monohaploid, and there should be heterozygosity whenever we look at the resulting assembly. And both of these have a haploid number of 11 chromosomes. Now, arabica, on the other hand, as I said, is a tetraploid. So it normally would have 4 copies of every chromosome. But the one that we used is a special accession, and it's a dihaploid. So this one has 22 chromosomes instead of the normal tetraploid number. So you can see here what a cartoon of what a dihaploid is. So with a tetraploid, you could have variation occurring between the homologous chromosomes, and you could also have variation occurring between the homologous chromosomes. With the dihaploid, any variation we see is only going to be coming from the 2 homologous chromosomes, so the 2 parental species essentially. So this is really nice for assembly purposes because this is essentially like assembling a very heterozygous individual instead of having variation coming from between the chromosomes and across the subgenomes. So the next thing that was really important that allowed us to do this was the availability of long sequencing reads. So we had initially attempted to assemble arabica with Illumina reads, and we were never able to get very good assemblies by using a Illumina. And part of the problem was the repeats in the genome were breaking contigs, and so our assemblies were never very good. So we set out after our Illumina attempts to do some assemblies with PacBio data. So you can see here the types of data that we had for our initial assemblies of canephora, eugenoides, and arabica. Arabica was the first one that we sequenced, so we have a little bit of older technology mixed in with that one. Arabica, we had about 56x coverage of the genome. Eugenoides was about 58, and canephora was about 77. We used a pretty standard assembly pipeline. So each of the genomes, as you saw, had greater than 50x genome coverage from PacBio sequence. Then we took that sequence and we did the standard self-correction and assembly. So depending on the genome, different assemblers produced different results. So I'll get to that more later. For canephora, we had some scaffolding and mate pair reads and also some back sequence. So we were able to do scaffolding with that, and then some gap filling with PB jelly and the PacBio reads. For arabica and eugenoides, since these were essentially heterozygous, we used Falcon Unzip to assemble those, and then we polished all of the assemblies with Illumina reads that we had from other sequencing projects from the same individuals. And then we took these and we sent them off to Dovetail for Hi-C, and then we took those and we filled them in with PB jelly in the original PacBio reads. So this is what our contigs look like. So this is before any further scaffolding or Hi-C was done. So we have the published canephora in the first column, so you can see what that one looked like. So that one is not sequenced with PacBio. And then we have two different canephora assemblies that we tried. One was with MHAP and the Celera assembler, and another was with Falcon. In this case, we found that the MHAP and Celera assembler produced a little bit better assembly. It was a bit longer, and N50 was a tiny bit higher. So that was the one that we used for our downstream analysis. Then we also had two different Eugenoides assemblies. Both were essentially ran with Falcon, but one used some DNA nexus-specific parameters, which performed a little bit better. So that was the assembly that we moved forward with there. And then we had three different arabica assemblies that were done with two different versions of Falcon. So one was an older version, one was a newer, and then we ran Falcon unzip on the assembly from 2016. So this is the assembly that we use for all further analyses here. Overall, you can see the canephora assembly seems to be performing the best, which is probably expected since this one is a monohaploid and should have essentially no variability. Eugenoides is slightly worse likely because of the heterozygosity issue. And then arabica, of course, is the most complicated one since this is our dihaploid. So this slide should look pretty familiar to you, and I think you've had a pretty good introduction to that, so I don't need to go into it in too much detail. But we took our coffee leaves that we had from our collaborators in France, and they were sent off to Dovetail for cross-linking. And then the libraries were created for the sequencing, and then Dovetail did their magic and got back to us with some really improved results. So you see here. So these are the results from the Hi-C scaffolding, and so basically what happened here, we sent canephora and arabica. We unfortunately didn't have any more Eugenoide samples to send to do Hi-C. So that one stayed the same. That's why it looks considerably worse than the other two. But for canephora, the Hi-C improved the scaffold N50 from 972 kb to 50 megabases, which was a 51-fold increase. And approximately 87% of the assembly is contained in 11 chromosomes. So that's really good because we should, the goal is to get to chromosome level, and we're nearly there. For the arabica assembly, since this genome again is a little bit more complicated, it wasn't quite as good, but still a very impressive improvement. So an N50 of 760 kb to 45.84 megabases. So this is a 60-fold increase over what we had sent them, the original contigs that had been sent to Dovetail. And so about 74% of this assembly is contained in 22 chromosomes. So this is very helpful for us since we want to do comparative genomics, and this will really allow us to do that. So then we wanted to look a bit more in these assemblies and see how they were performing. So we used BUSCO as a metric. So these are genes that should be present in single copy in most plant species. And so we looked through our assemblies and then also through Arabidopsis and tomato to have a comparison to some genomes that we know were good to see how many of these we were able to find. So we have the published canephora there as well. And you can see that our canephora assembly is a little bit better in that it has a couple more single copy genes and a few less genes missing compared to the published. Eugenoides performs slightly worse, likely due to the heterozygosity. And arabica, of course, is the dihaploid. So here what we're detecting is the homologous gene copies here. So that's why you see so many that are duplicated. So that's expected. We also compared the canephora genome to the Eugenoides genome, and we were expecting these to be pretty similar because previous work has been done that's found that they're quite similar. So you can see the 11 chromosomes from canephora on the x-axis and the 11 chromosomes from eugenoides on the y-axis. And these are just dot plots that show the comparison on a per chromosome basis. And there's pretty good collinearity between the two genomes as expected. So one other thing that we did, and this we're actually still in the process of doing, we have optical maps and genetic maps and a lot of short read sequencing for arabica and the two parental genomes. So we've taken the arabica genome and we're assigning the homologs so that we can tell which parental genome that they're most similar to. And then also we're doing a bit of checking of the assemblies. So since we have the optical maps, we're able to see if there have been any misassemblies or if things are looking pretty good overall. And so far from the work we've done, things are looking pretty agreeable. So now that we have these genomes, of course the more interesting thing is knowing about the genes and finding them in the genome so further work can be done there. So we have iso-seq data from PacBio, which has been really great for annotation since it gives you basically the whole transcript length. And we've used that as well as ESTs and RNA-seq to run the maker pipeline. So we trained the ab initio predictors, Augustus and SNAP, and then we've used all of this expression data to predict the gene models in the genome. And then these, we're predicting a functional prediction based on BLAST to different databases and also InterProScan to associate the Go terms to these. We also set up a Web Apollo instance, which has been really helpful for the manual curation of genes. So this can also help you improve your training set too for Augustus and other predictors. So this is just showing an example of a gene model that we've set up in that system that then people that are part of the consortium have been able to go in and find their favorite genes, so things maybe involved in caffeine biosynthesis and other pathways to make sure that they've been correctly annotated. And if not, then they can actually manually fix them. So this will help improve our gene models in the long term. So far, this is what our annotations are looking like. So this is pretty comparable to the published canephora. I've included tomato and arabidopsis as well. And so this is still a little bit of a work in progress to sort of fine-tune these. This is probably one of the more difficult things in genome assembly. And producing a good genome as a resource is really getting the gene models as good as you can. And as you can see, arabica has roughly twice the number of genes as canephora and eugenoides. So this is another indication that it looks like we're able to detect both homologs of the two from the two parental genomes in the assembly. This is, so just looking at BUSCO again, this time we're looking at the actual annotation. So before we were looking for these genes in the genome, now we're looking at the annotations to see if they're in the protein models that we've predicted. So if you look at canephora, so our version is the 1.4. It's performing pretty well based on the BUSCO metric. So this annotation is pretty set. But eugenoides, well, you saw the genome is still missing some genes, so we're just not really going to be able to detect some of these because they didn't assemble very well for some reason or another. And again, you're seeing an increase in duplicated genes in arabica, which would be expected. So the next step, because a genome by itself isn't really so interesting, is to do some more work to look at some of the genetic basis of interesting natural variation that I was explaining previously. So trying to find genes that might be involved in pathogen resistance, insect resistance, things that would be useful in breeding. And also we're interested in evolution of coffee, so we'll be using our genomes to study that. And also in our lab, we have the tomato database called Soul Genomics. So one of the big things we do there is develop toolkits for breeders to use. So you can go there and, so this example is tomato, of course, but you can go there and you can find things like markers for breeding, various phenotypes, genes that are involved in interesting characteristics. You can identify QTLs using tools there. So the goal is to get information from coffee into a similar resource so that breeders and the public can access this. And then the goal is to help improve coffee. So I'd like to acknowledge the Arabica Coffee Genome Consortium, which is a pretty large group of people from 9 countries, 18 institutions, and 53 researchers, and also a big thanks to Dovetail for allowing me to speak today.
Well, thank you, Dr. Ho and Dr. Strickler, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask Dr. Ho and Dr. Strickler, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, our first question is, how long does it typically take to go from tissue to delivered assembly? And this question is for Dr. Ho.
Sure. Thanks, Judy. In general, depends on what you need to be done. But in general, it takes about 12 weeks from the day that we receive your sample up to the day that we deliver the report and the final assembly for you. But of course, if you need to do de novo assembly, which we also provide as a service, it will just take a little bit longer. But in general, about 12 weeks.
And before we move to the next question, I want to advise that a series of poll questions will appear on your screen during the Q&A. You may select your answers, then close the poll questions by clicking on the X in the right corner. Okay, this next question is for Dr. Strickler. What percentage of the genome was masked for the repeats, and how did you avoid masking something that you should not?
Sure. So I think in the end, about 60% of the genomes were masked whenever you look at the 3 different genomes that we worked on. So they're pretty comparable since they're closely related. And the way we did the repeat masking was we first looked for a de novo repeat. So we used Repeat Scout and Repeat Modeler. And then we filtered those repeats for known protein domains, because if you don't do that, what ends up happening is sometimes you can mask out things that are members of gene families. So we filtered our repeats and removed things that were known domains, and then we used that set for our repeat masking. So that did a pretty good job of not masking things that it shouldn't do. I mean, nothing's ever 100% perfect, but it was as close as we could get.
Okay, this next one's for Dr. Ho. We are looking into getting an assembly, but working on which approach to use. Do you provide consultation and options that address our concerns about price and quality?
Sure, absolutely. So to be honest, we will not start the project until we have a good idea of what your expectation on the genome. Because we understand that no 2 genomes are the same. They come with their own complexity. They're all different sizes. As Susie has shared with us about coffee. So we're definitely here, and we'll be here to help you and try to decide what's the best strategy forward for your genome size, for your given complexity, and also a price range that would match what you need to do downstream. So absolutely, we're here for you.
Okay, and this next one is for you too. Could we get to whole chromosomes with a de novo and Hi-C?
I would love to say yes, but I guess we have to be realistic. First of all, chromosomal level assemblies depend very highly on the input assembly, right? Of course, from what we have observed from our Hi-C rice, if your input assembly is close to in megabases, that's where the Hi-C library will actually fly, and it is able to stitch things into close to chromosomal or chromosomal level. And which is why I'd like to illustrate, discuss a little bit more about why we actually have Chicago as well, because if your input assembly is not that big, or the N50 is not that great, you need some kind of intermediate scale library data to actually bring it up to where Hi-C can actually bring it down to chromosomal. Yep, so that's the answer.
Dr. Strickler, this next one is for you. UC Davis announced earlier this year that they had sequenced the arabica gene. How does that work relate to the work of the ACGC, and what were the main differences in their approach than your consortiums?
Sure. So yeah, it's completely different groups, and there are actually a number of groups throughout the world that are currently active in sequencing a particular accession of arabica or another. To my knowledge, we're the only group that's doing a dihaploid, so that should give us a bit of strength because it should be a bit easier to assemble. But it'll be interesting to see how they all come together and to have a number of different accessions so we can look and compare, do some comparative genomics with what comes out of these different consortiums.
Dr. Ho, this one is for you, and I'm not sure which slide it applied to. Is this technique applicable to plants only?
Well, absolutely, we can. Well, plants is, to be honest, it's one of the most challenging things because, as Susie has mentioned, there's all these homologous regions. We have had success with mammals as well, as you can see from the slides, fish and reptiles. They are generally a lot easier to assemble and put into chromosomal level assemblies. So absolutely, yes.
Okay. And this next one is for you. For a tetraploid genome, let's see, let me just read this over once before I ask it to you. For a tetraploid genome for which you do not have dihaploids, are you open to try it?
Sure, of course. We have some clients that have approached us with exactly the same problem. We understand that it's always not easy to get dihaploids. So we are most happy to have a discussion. And the most important thing is to find a viable solution for our clients, which is why we're here.
Okay. And Dr. Ho, can assemblies generated by any means be scaffolded with Chicago and or Hi-C? And if so, what qualities do you look for in input assemblies?
Sure, yes. Most of so as far as we have gone through, yes, any assemblies, be it PacBio, whether you've done a shotgun, we can scaffold it with Hi-C and Chicago. Yep.
Okay, and this next one is for you. Can you go straight from a de novo assembly to Hi-C, and when would you use Chicago?
Sure, of course. So the difference between Chicago and Hi-C is the characteristics of the data. So Chicago is more intermediate scale. So if your N50 is in kilobases, we would strongly recommend you go to a Chicago. That will hopefully bring it up to megabase level N50. And once you reach that, we should be able to go up to use Hi-C, where we can bridge longer scaffolds. So it depends on the N50 size of if you're assembling.
Okay, and our next question is this technology applicable in induced polyploid species? From our experience, it seems to work pretty well. I'm most happy to connect with you and see how we can work for it. And while we're waiting for additional questions to come in, I would like to once again thank Drs. Ho and Strickler for their presentations. Do you have any final comments? No, from my side. Thank you everyone for being here with us. Okay, and I have another question for you. Sorry, Dr. Strickler, before you answer, can I throw another question at you?
Sure.
Have you tried PacBio and BioNano Assembly as an input for Hi-C assembler?
Is that a question for Xuan?
Oh, I think that one's for you. Is that for yes, Dr. Ho, or shall we save that for after the presentation? Maybe
sure, no problem.
email response.
Yeah, I can answer that. BioNano assembly, so the data coming from BioNano is also considered kind of intermediate range data up to 100 kbs. Yes, we can definitely go ahead with Hi-C data on top of BioNano.
Thank you. And Dr. Strickler, we will look forward to your closing comments.
Yes, thanks a lot for having me and giving me the opportunity to speak here.
Well, thank you. Thank you both once again. I would also like to thank our sponsor, Dovetail Genomics, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through March 20th, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.